All right, this is the second part of the cell membrane PowerPoint information. This is kind of a bonus video. Because last one was so short, I want to get this done for Wednesday. You guys can do it, I know. And we'll probably be able to get a whole unit done in a week, which will be glorious. And I also probably won't have a test on this because it is all review. You'll just have a quiz on this on Wednesday. Also, I got new glasses. Well, they're not new. I should have been wearing glasses this whole time and I lost them. So I'm excited, and I think everyone who is also on the road is safer and happier because I'm driving with glasses now, which I should have been doing for basically a year. Adulthood. All right, so this is all transport. Plasma membrane, we talked about it, you know it. This, you know. So we're just gonna, I would in your notes do like numbering it. We're just gonna go through the types of transport across the membranes. Passive diffusion, we already know. This requires no energy to move, and it moves from high to low concentration, basically from where there's less space to where there's more space for it to hang out. This image is just showing how the red molecule moves towards the A side because there's more space for it. Diffusion. There's a video, but we're not going to watch it. You can also make this chart. Pause, put this chart in your notes. These on the left are the types of transport we're looking at. Facilitated diffusion. It requires a transport molecule, but still doesn't require ATP or energy. The driving force, again, is still moving from high to low concentration, or entropy, right? Talking about disorder. It's just moving from high to low concentration. Creates a concentration gradient. It just is too big to move across the membrane through diffusion. Yep, this is just looking at how it moves, right? Goes through that little thing. It's just too big to move across through diffusion. Again, same things. Also examples, I'm not going to talk about those, but I would write them down in your notes. Active transport requires a protein, also requires energy. It can also move against the concentration gradient, so it pushes stuff into a already filled up space. This one is most commonly seen in the sodium-potassium channels when we're making a concentration gradient for a nerve impulse. Active transport, a little GIF image that's not going to play, so we'll skip it. There's a specific type of active transport called an electrogenic pump. And it is a pump that pushes ions against the concentration gradient, and it creates a charged membrane. So we see this one specifically when we are conducting an impulse, right? We determine the charge by pushing more sodium or potassium, which are positively charged ions. This is a type of sometimes passive transport, but usually active transport. Okay? Osmosis, type of diffusion, specifically the diffusion of water, moves from high to low concentration. High to low, yep. This is the most common image that's used to show how water moves. It also makes us think about the terms hyper, hypo, and isotonic. And so I would think back and remember what those terms are. I'm not going to cover them right now. I will in class if you ask me to. But here's the slide, so you can pause it and take a look at it if you want. So we have endocytosis. Endocytosis is a type of active transport. It's the movement of larger particles, bigger than just moving through a transport protein. And there are three types of endocytosis: phagocytosis, pinocytosis, and receptor-mediated endocytosis. But for all three of these, it makes a vesicle. So if you remember how with the Golgi apparatus or the endoplasmic reticulum, it creates this like little bubble pocket and then it pinches off with something inside of it, that's what's happening. Phagocytosis is cell eating. So it's going to engulf larger particles into a vesicle and then it'll transport it where it needs to go. Pinocytosis is cell drinking. It takes dissolved molecules that are brought into the cell. It also engulfs liquid. So phagocytosis is mostly what we consider food or large chunks of stuff. Penocytosis is going to be liquid with dissolved particles. The last type is receptor-mediated endocytosis. Basically, receptors on the outside of the cell membrane allow for the attachment of a particular molecule. And then when a certain number of those receptor sites are filled, endocytosis, or the engulfing of it, occurs. So the receptor-mediated means it needs a certain amount of receptors to be set off before it engulfs what it needs to engulf. Exocytosis, so not a type of endocytosis, but a type of active transport, is the opposite. Basically, it is the expelling or the removal of things. So it's just the other process. The first three were things coming into the cell. This is material being released from the cell in the same manner. Obviously, the far left images are going to be the best images that show you what that looks like. This is the final table of the things we talked about. I would recommend, if nothing else, putting this table in your notes and making sure you understand these types of transport. Here's the blank one if you want to take a picture of it or copy it or do whatever you want. And that's it. 2 super short videos. These are for Wednesday, and you will have a quiz on them.